University in the United States. So I hope you all enjoyed the keynote by Dr. Hatton Howard yesterday and feel inspired to know a little more about microbial metagenomics analysis. So the title of my talk is Reconstructing 16S ribosome RNA genes from metagenomic data. Here is the outline. So first, I will give you a very brief background about metagenomics and 16S ribosome RNA. After that, I will explain how we reconstruct 16S ribosome RNA genes from metagenomic data. And finally, I will present the results. In short, our tool can recover about 25% more ribosome RNA genes than the next best tool. You all know microbes. Good germs help us digest food. Bad germs cause disease. So they are the oldest life form on Earth, and they live everywhere. However, our understanding about microbes is very limited, partly because there is a very small percentage of them can be cultured and sequenced in labs. Fortunately, with the development of metagenomic technology and next-generation sequencing technology, people are able to sequence the DNA contents from environmental samples without cultivation. So now scientists can study both the culturable and the non-culturable species in a more comprehensive way. So now I'm going to show you the major steps in metagenomic sequencing. So first, the samples will be collected from different environmental samples, such as from soil, from ocean water, from human gut. And then the samples will go through some filtration, usually size selection, and then the library will be constructed. And here you will see the computer that represents the high-throughput sequencing machine. So the output of the sequencing machine contains a large number of short reads. That will be the input to the downstream bioinformatics analysis. That's also the input to my research problem. So let's take a closer look at the next generation sequencing step. Okay, so here I have two figures. On the left side, that shows the sequencing of one reference genome. Usually, this genome is too long for the sequencing machine. So in the first step, this reference genome will be broken into shorter fragments. And then these fragments will go through the sequencing machine. You can either do single-end sequencing or paired-end sequencing. In the paired-end sequencing, both ends of this fragment will be sequenced. This is the input to our algorithm. So our algorithm is optimized for paired-end sequencing. We need to use that information to distinguish different microbial species. And this is for just one genome. In metagenomic sample, we have a variety of microbial species of different abundance. So as a result, the smaller figure you can see, different colors represent different species. So we have lots of short reads coming from a variety of species. And of course, at the beginning, for each read, you don't know its taxonomic information. There are lots of research questions asked for different metagenomic studies, but there are two questions routinely asked for each study. The first one is, who is out there? This is a composition analysis. We want to know what species are living in different samples. For example, in human microbial project, metagenomic sequencing is conducted at different body sites across different people. We want to know whether there is a core microbial across different body sites and across different people. The second question is the functional analysis. So in this talk, we focus on the first question. And the major method for phylogenetic analysis or composition analysis is still based on 16S ribosome RNA analysis. So now let's take a look at the gene 16S ribosome RNA. So this gene is a very good marker gene for phylogenetic analysis. First, it exists in almost all bacteria and archaea species. Second, it has a very nice structure for phylogenetic analysis. It is a patchwork of highly conserved regions and also species-specific regions. So those highly conserved regions can be used for primer design, and those variable regions are good for species-specific applications. That is to identify the species information from those regions. Okay. However, so now the major method is based on comparative analysis. For example, if you have a query 16S ribosome RNA gene, you can compare your query sequence with the characterized 16S ribosome RNA genes from a reference database, such as from the RDP database, Ribosome Database Project. It contains hundreds of thousands of characterized ribosome RNA genes. So based on the similarity, you can infer the taxonomic information of a sequence. However, this method cannot be directly applied to metagenomic data because it works better for long sequences or near complete genes rather than those short reads in metagenomic data. So there is a need to reconstruct the genes from those short reads first. This is basically what I'm going to solve or talk in this presentation. So the input to our problem contains all the reads in metagenomic data, and the output will be near complete 16S ribosome RNA genes or the context. So sometimes the sequence coverage is so low, you cannot recover the whole genes. In that case, we will try to give you context. And this is not a trivial computational problem. There are several computational challenges. First, data size is very big. Second, the reads are short and contain errors. And we do not usually have reference genome for all those genes because we are expected to recover novel species. So you cannot just do read mapping. And also, those highly conserved regions between 16S ribosome RNA genes could make the graph very complicated. So I'm going to give you an intuitive idea about why this problem is very difficult. So here, in this slide, you see three figures, the overlap graph. In each overlap graph, every rectangle represents a node that is basically a read. And if two reads share a significant overlap, then that overlap is represented by a connection, an edge in the overlap graph. From the left side, we can see the overlap graph for one species, two species, and three species. You can see with the increase of the number of species, this overlap graph becomes very complicated. So our goal is, once you have the overlap graph for all the reads, we want to traverse the graph and find out the paths that correspond to the correct 16S ribosome RNA genes. So here I already highlight the gene paths. One is, for example, in the middle, we have two genes that are highlighted by red and blue, and in the right, we have three highlighted paths. So you can see this graph becomes very tangled, okay, with only just the three species. And how can you figure out the correct paths? That is not a trivial problem. And of course, you might say, okay, how about we just use generic de novo metagenomic assembly? So I will show you later on that those programs are not optimized for 16S ribosome RNA assembly. This is outline. This is overview of our method. I will only highlight the most important parts in this pipeline. So first, we want to solve the scalability problem. For all the input data, all these reads here, in the first step, we're going to do a filtration based on homology search. We want to recruit or identify all the 16S ribosome RNA reads. Those are the reads likely to be sequenced from 16S ribosome RNA. Usually, after this step, only a small percentage of the reads will be kept for downstream analysis. So those reads, we'll use those reads to construct an overlap graph. And overlap graph construction is usually a very expensive process, so I will briefly explain that. And once we have this overlap graph, we are going to do the pathfinding. Here, we will take advantage of the pair-end information to distinguish different species. And finally, we are going to do the scaffolding. So here, we're going to use a model as a reference, which is a probabilistic model, so that we map all the contexts to the model, and then we know the orientation and order of those contexts. Okay. So first, we're going to recruit all the 16S ribosome RNA reads based on homology search. When you look at the structure, you can see some regions are highly variable, so they do not always have high sequence conservation. In order to do the homology search with higher sensitivity, we use this probabilistic profile model, which encodes both the sequence and the secondary structure conservation. So this is secondary structure of 16S ribosome RNA genes. Okay, even when the sequence conservation is low, they could have very high secondary structure conservation. So we use that homology search to align all the reads to this model to recruit the reads that are sequenced from 16S ribosome RNA genes. And after that, we're going to generate the overlap graph. You need to do the all against all comparison to find out all the overlaps between reads. That's very expensive. Quadratic, okay. So fortunately, there are some advanced data structures available for overlap finding. So we're using read joiner. It gave us almost a linear time implementation of this overlap graph. After we build the overlap graph, the initial graph is very complicated. So as shown here, we have 3 species. You can see there are still edges connecting different species. We want to break those edges. In addition, there are sequencing errors. There are conserved regions, that is the chimeric nodes, okay, shared between different species. So we use existing error correction methods, such as those based on topology search. You can remove the tips, bubbles. And also, the novel part here is we will remove the bad edge. The bad edge means the edge connect different species using some phylogenetic analysis. We use Bayes classifier, naive Bayes classifier from RDP to figure out, okay, what are the possible genus for each node. If 2 nodes, they do not share any common genus, we're going to break that edge. And of course, initially when you have very short reads, this classification does not work very well. So that's why this graph pruning process is iterative. When we have short reads, sometimes it can be combined as a longer contig. And then we are going to apply this process again until the graph cannot be simplified anymore. Next, once we simplify the graph, we will do pathfinding. We are trying to find the path corresponding to each 16S ribosome RNA gene. This is a very difficult step because with the highly conserved regions between different sequences, we could get chimeric sequences. That is a sequence containing different parts of different 16S ribosome RNA genes. So the major method we use here is paired end information. So I'm going to use this example to explain this idea here. So this is a very small overlap graph, and the dashed line represents the paired end reads shared between nodes. Okay. So for example, we already have this path starting from N1 and ending at N6. Now we need to decide, okay, what's the next step? We could go to N7 or N8. So in this case, we check the number of paired ends support for these 2 paths. Which one has higher support, we will choose the next node to traverse to. So in this case, we designed a scoring function, basically to scoring all the paired end reads for all the nodes along each path, and the one with higher score will be chosen. So in that case, we are going to choose N7. Okay, so now I'm going to present the experimental results. We did several experiments. Here I'm only going to present the results on a mock community data. We are going to benchmark our tool with several other tools. First one is called eMERGE, which is probably the state-of-the-art tool for ribosome RNA gene reconstruction from metagenomic data. It uses a read mapping-based strategy, so it needs some reference sequences. And then we will benchmark our tool with 2 other de novo assembly tools. One is IDBA-UD, the other is MetaVelvet. So they are optimized for metagenomic assembly. Their goal is to recover all the species, all the genomes of different species. And those tools, they do not need any reference sequence. Okay, so this is basically the data. It's a real metagenomic data, but the community is synthetic. It contains 64 species from bacteria and archaea. It covers 5 genera, and the read is paired end. The length is roughly 101 nucleotides. So first, let's look at the performance of homology search or the filtration step. Recall that in this pipeline, in this pipeline, the first step is to classify all the reads that are sequenced for 16S. So we want to see how good we are. And this table shows the sensitivity and specificity. So basically, we consider two models. The first one is the first training set excludes all the genes in this multi-community data, 16S genes. The second model excludes all the genes in the 50 genera because we want to see whether we can recruit novel 16S reads. As you can see, both of them have very high sensitivity and specificity, but the running time indeed is high, about 12 hours. And now let's look at the performance about the final results. That's how we recover the 16S ribosomal genes. And again, we consider two types of input. The first one contains all the original reads. The second one contains only the real 16S ribosomal genes. We want to see both performance. As you can see, our results, we have much higher sensitivity and the specificity. And the running time, the bottleneck is in that homology search stage, not in this assembly stage. And in merge, when we exclude all the genes from the 50 genera, it only covers less than half of these genes. And similarly, when we use only the real 16S ribosomal genes, our performance is better, but still we have the highest sensitivity and the specificity. So finally, I want to thank the NSF support for this project, and we do have some future work. And for more details, please read our paper. Thank you very much for your attention.
Thank you for this nice talk. We have time for questions. For questions, I will invite you to go online and take the microphone there. Yeah, please.
Hi. Yeah. Nice talk. So, if you're sequencing the entire metagenome, what's the the core motivation to use a 16S RNA? I mean, you could also use the housekeeping genes or the core genomes or the pan genome to figure out or possibly if the goal is phylogeny, disambiguate the
so using the whole genomes will be good for phylogenetic analysis. However, for complicated metagenomic data, it's very hard to recover the genomes, such as the soil metagenomic data. It's very difficult. For example, you can apply IDBA, UE, try to recover all the genomes, but it's very computationally challenging, especially the memory. And also the accuracy is not that satisfactory.
Any more questions? So if no, let's thanks again the speaker.